avoid them. I'd like to thank the organizers for inviting me here to share in the celebration of Govind and his work. And Govind, as you know, has been a colleague of mine at MIT for a quarter of a century. And I'm grateful to him for having inspired many of us to focus on fundamental problems and to do so in a way that would create a solid foundation for future work. I'm going to focus my talk today on programming a cell state. And what you see in this slide is an abbreviated version of vertebrate development. And it's meant to simply point out that not only are there a wide variety of cell types, perhaps 200 different cell types in the adult vertebrate, but there's a process by which they're generated. And we have yet to understand much more than the very basics of how these processes are controlled. What I'd like to do is to try and address what we think are some of the key regulators of cell state, how they control cell state, and how reprogramming is accomplished based on this. And to do this, we need to focus initially on one cell type, and embryonic stem cells provide us with a great model cell to study some of these problems. They have two key properties: pluripotency, the ability to differentiate into almost any cell type, and self-renewal, the property that makes them valuable to biologists who need lots of cells to study. So the first question: what are the key regulators of embryonic cell state? We've known this for a little while. They are transcription factors. 4 of them are OCT4, SOX2, NANOG, and TCF3. The first 3 have been known for about a decade to be essential for the 2 key features, pluripotency and self-renewal. OCT4 and SOX2 form a heterodimer. NANOG works with a variety of other partners. And TCF3 is in this TCF-left family, which will bind beta-catenin upon Wnt signaling, and it's known in ES cells to be one of the factors that mediates Wnt signaling. And if we weren't persuaded that these transcription factors play fundamental roles by the genetic data that's accumulated over the last decade, the really earth-shattering work of Shinya Yamanaka has persuaded most of us that in fact these factors have the property of being able to program an embryonic cell-like state. This work's been duplicated in many labs. It's been shown both in Shinya's lab and in labs here to generate iPS cells from human fibroblasts. And these cells have the ability to in fact generate embryos. It's not yet clear whether they're absolutely identical to embryonic stem cells, but I think the accumulating evidence is that they in fact are almost indistinguishable from embryonic stem cells, both from the perspective of the biological properties they exhibit and from the perspective of molecular characterization of their genomic behaviors. Now, it's not just transcription factors that play key roles in regulating cell state. So it's known for ES cells that there are many other components that are necessary for maintenance of the embryonic stem cell state. These include polycomb group proteins, which we'll talk about a bit. These are chromatin regulators that modify histones. MicroRNAs that are preferentially expressed in ES cells play a role in maintenance of their state. And there are a variety of signaling pathways that are functional in ES cells and whose functions are critical for the maintenance of that state. So how do these regulators control cell state? Many of them have their ultimate behavior in transcription, so we need to talk a bit about this. Here's a conventional model for how gene transcription is regulated. The transcription apparatus is recruited by DNA binding activators to promoters. There are events that lead that lead RNA polymerase to escape the promoter and make RNA. At inactive genes, there are also DNA binding factors, specificity factors that prevent these events from occurring. And in our work and the work of many others, it's become evident that chromatin modifying enzymes work in a manner that's associated with these transcription apparatus. So for example, the trithorax group complexes are recruited to sites with transcription initiation to modify histone H3 on lysine 4. And as the transcription apparatus moves further down the gene, then a set2 domain containing histone methyltransferase modifies other residues on local nucleosomes. There are histone modifying apparatuses that are also associated with genes that are repressed. That model has been modified somewhat in that we see in vertebrate cells, whether it's human ES cells, differentiated cells, or murine cells, that there's a substantial population of genes that experience transcription initiation but not productive elongation. And those genes in embryonic stem cells include many of the lineage-specific developmental regulators. They encode many of the lineage-specific developmental regulators, which are poised for transcription during later stages of development. Now, they're poised, and at least a subset of them are repressed in part by the action of these polychromate group complexes that catalyze histone H3 lysine 27 methylation. A technique that was mentioned earlier by Lloyd has allowed us to look across the genome at the behavior of many of these transcription factors, chromatin regulators, and other kinds of DNA-associated proteins. It's described up here in brief. It's been very valuable for us in nucleating models for how these transcription factors and other regulators control the genome, and it effectively uses cross-linking of proteins to DNA, shearing the DNA, the isolation of those cross-linked protein DNA complexes with antibodies, and then determination of the enriched DNA fragments by methods like DNA microarray or more recently, DNA sequencing. And when you use these methods, you get results that look much like what I've shown here for in murine embryonic stem cells. The gene that encodes OCT4, that's POU5F1. Here's a gene model. These are two well-characterized enhancers for the OCT4 gene. And what you can see is, as you might expect, the DNA binding transcription factors that are known to regulate OCT4 are bound to the well-characterized enhancers. So these four transcription factors. RNA polymerase II is bound to the core promoter, and you can see signals that appear over the enhancers. I think they appear over there because the transcription factors are bound to components of the initiation complex, and formaldehyde cross-linking leads to protein-protein as well as protein-DNA cross-links. You can see histone modifications that are associated with gene activity across that gene, and if you sequence RNA, but you don't sequence it too deeply, you see the stable exonic sequences. If you sequence it more deeply, you begin to see sequences that come from the exonic intron sequences. And we can take this data for the entire genome and use it to nucleate models of regulatory control, and that's what this model is up here. And you can't see the names of these genes, in part because the names of them are not important, and this just summarizes what's going on for the entire genome. So there are for OCT4, SOX2, NOG, and TCF3, some 3,000 target genes, and I'm just displaying a few of them. I'm displaying a few of them for the purpose of telling you that there are a few key features that I'd like you to remember. One feature is that these master transcription factors, these transcription factors that can not just program ES cells, but reprogram somatic cells, bind to and regulate their own promoters as a group. They're a group of drinking buddies that work together at each of their own promoters. And we think that this motif promotes what is called a bistable state. If these transcription factors in blue are present, and the proteins are present, they positively regulate their own gene expression. This is a motif that is jump-started during reprogramming. We'll talk about that later. The key factors, OCT4 and the NOG, when you stimulate ES cells to differentiate, or when the inner cell mass cells and the blastocysts are stimulated to differentiate, are rapidly degraded. And that breaks up that circuitry, allowing other master regulators of differentiated cells to take over. A second feature I'd like you to remember is that they're actually, the target genes of these regulators fall into two classes, active and repressed. The active genes, as you might expect, include most of the things necessary to run the cell and allow self-renewal. And we know that the binding targets of OCT4 are functional because when we can knock down OCT4 and show that that causes loss of expression of these genes. And the representations up here include, in boxes, genes for messenger RNAs and in proteins, and also microRNAs in purple. That second class of genes I mentioned, the set that are repressed, are interesting in that they include essentially all the lineage-specific developmental regulators. And that includes protein-coding genes as well as microRNAs that are known to have roles in differentiated cells. And this is important because were you to turn on some of these regulators, which are master regulators of specific cell types, you would lose pluripotency. So we think that the way that these master transcription factors, OCT4, SOX2, and TCF3, control pluripotency is in effect by ensuring that the master regulators of differentiated cell types remain off. It's polyclonal group proteins that are contributing to the repression of those genes. The microRNA genes that are preferentially expressed in ES cells are occupied by these master transcription factors, and they form what are called feedforward loops. In fact, incoherent feedforward loops is the terminology that's used by the systems biologists. And that's where the transcription factors are positively regulating the microRNA genes, at the same time positively regulating a group of protein coding genes that are essential for ES cell state. The microRNAs seem to tune down the messages for these proteins. And one of the reasons we think this is important is because during differentiation, when you turn off OCT4 and the NOG, you'll also need to rid yourself of messages for transcripts that are no longer essential for that cell state. And so, in fact, these microRNAs look like they have the property of continuing to cause embryonic stem cells to remain poised for differentiation. Dicer knockouts, for example, lead ES cells to have a much more stable phenotype in culture. They tend not to differentiate. The thing I want you to remember about signaling is that for all the developmentally important signaling pathways, the terminal transcription factor that's the effector for those signaling pathways actually comes to the bar with the master transcription factors at each of the promoters where they operate. So what we and others have now seen is that TCF3, STAT3, the various SMADs that are active at the end of the active nodal pathways, bind promoters together with these transcription factors. So these developmental signals for these pathways are actually brought right to the business end that is the set of 3,000 promoters where the master regulators operate. We wanted to look for new regulators of ESL state, and so Mike Kagi and Jamie Newman, Steve Bilodeau in my lab set up a screen using murine embryonic stem cells and a short hairpin RNA library that was produced by David Sabatini in collaboration with the Broad Institute to look for transcription factors or coactivators or chromatin regulators, signaling proteins, whose effect would be to knock down OCT4, which is our key, it's a key marker for pluripotency. And I'm not going to go through all the data, but I do want to point out two things. One is that we got all the transcription factors, coactivators, chromatin regulators, and signaling components that were previously known. So that was very reassuring. But two essential protein complexes came up that intrigued us. One is the mediator. Most of the subunits of the mediator were targeted by this pathway and have the effect of knocking down ESL state. And a complex that was initially described as being important in holding sister chromatids together during mitosis. So I'm going to tell you a little bit more about these two protein complexes, in part because I know they are studied by a number of labs at the University of Wisconsin. So the mediator was of interest to me because the first decade or so that I worked at MIT, I worked on this complex in yeast, so it's quite familiar to me. And it was a surprise that it was such a profound target of this screen, because I just thought of it as a component of the general transcription apparatus in most cells, and I still do. But it looks like when you knock it down, it has a profound effect on the ESL state. So transcription factors recruit the mediator by binding to specific subunits. We suspect that this contributes to DNA loop formation where enhancers are brought in proximity to the core promoter. We know in humans that mutations in various subunits cause a whole variety of developmental diseases. So we're going back now to ChIP-Seq, and again, these are the transcription factors at the gene that encodes OCT4. And when we do mediator ChIP-Seq experiments with a variety of subunits, we see what is summarized here for one of those subunits, MED12. And that is exactly what we expect. It's found right over the two enhancers, so the DNA binding transcription factors are binding to the mediator, and it's found over the core promoter, where you find RNA polymerase and tau binding protein. So here's ChIP-Seq for TBP. And the data suggests to us that what is happening is exactly what we would expect from a tremendous amount of work in bacteriophage, prokaryotes, and a more limited amount of work in eukaryotes, which is that a loop is being formed either transiently or not so transiently between these enhancers and TBP. We're excited about this data because one of the biggest problems we have with this kind of technology is associating transcription factor binding to a region of the genome with a specific gene. That assignment is ambiguous and is typically done by proximity, but enhancers can be tens or hundreds of kb away from the gene. And this helps us create models for which enhancers are operating with which promoters. But we came to think that maybe the looping part of this was the most important part of the result of this experiment, because the knockdown of cohesin subunits would phenocopy the knockdown of either mediator or OCT4 itself. And cohesin is a multi-subunit complex. It forms rings. It's thought to form rings around two DNA molecules. It binds to a DNA binding factor called CT-Seq, which up until now is thought to have been a factor that binds insulators. And in fact, I'll show you, the Genome-Wide data says it does bind, that Cohesin binds to all the CTCF sites in murine embryonic stem cells. Cohesin mutations cause a variety of developmental diseases, but not mitotic diseases or mitotic defects. And that's of interest to us because, again, Cohesin was described first by Kim Naismith and others as key to the regulation of the metaphase-anaphase transition, and was only recently implicated in transcriptional control. So now I'm going to tell you something that I think is still in the realm of speculation, but I think it's quite exciting. So here again, we're focused on the gene that encodes ONC4. Here are 4, and I'm just showing you 3 of the master regulators bound to the 2 enhancers. Again, Pol Mediator, Pol2, TVP. And SMC1A is one of several subunits we profiled, so this is what we see for the other subunits as well. And CTCF, which is bound to some 10,000 sites across the genome. So now CTCF, here are the DNA binding motifs for CTCF, and in fact, as predicted, Cohesin is binding to CTCF sites. But what surprised us is, and this example is duplicated across the genome, it's bound wherever OCT4 and SOX2 are bound. And it's occupying, it would appear to occupy the core promoter sites as well. So what we think is going on here is that the enhancer is looping around to connect with the transcription initiation apparatus, and that is being stabilized in some way by Cohesin. And we think that's contributing to positive gene control because Cohesin is found exhibiting this behavior at all the active genes in murine embryonic stem cells, and not at the inactive genes in those cells. So here's one of many models you could imagine by which the transcription factor is a recruiting mediator, which is associated with the general transcription initiation apparatus, and Cohesin may be stabilizing that structure. So if we think this is some of how the program of embryonic stem cells is established and maintained, how is reprogramming accomplished? And the model that Rudolf Janisch and I propose and continue to believe is that when you introduce into cells, by any mechanism, exogenous genes that encode OCT4, NANOG, SOX2, and proliferation factors, MEK and KLF4, what you do is you begin to find cells that, as they're replicating under the influence of MEK and KLF4, you find cells where one or more alleles of the genes encoding the endogenous factors is open for business. And with time, what happens is that one allele and then the other becomes demethylated, probably passively on the DNA, and ultimately acquires the characteristics of an active promoter structure, which is histone H3K4 trimethylation and demethylation of CpGs in those promoters. In the early experiments, the retroviruses that were introducing these exogenous factors would become silenced, and the expression of the endogenous genes, having been jump-started, would auto-regulate their own expression. So that's, in a nutshell, how we think IPS reprogramming is likely to work. And we think the reason it takes so long is that you need multiple rounds of DNA replication in order to acquire this passive and stochastic and accidental loss of DNA methylation and of histone methylation that's repressing transcription. So there, as you'll hear more from Jamie Thompson a little later, what the field of regenerative medicine imagines is that individuals can have skin biopsies. The fibroblasts and other cells from those biopsies can be reprogrammed by defined factors. And in every case, these are transcription factors we know of. And that these patient-specific iPS cells or ES cells can then be used either for the study of specific disease problems or, in fact, to generate cells that are genetically matched and therefore immunologically compatible with patients. I think one of the reasons I wanted to show this slide is that I think that as we come to understand the transcription factors that are the master regulators of various cell types, we'll be able to circumvent this rather lengthy procedure and actually reprogram fibroblasts from patients directly into the cell type of interest. And it'll be, in fact, there are this week papers in Nature describing reprogramming of cells into brown fat cells, maybe, of clinical use. So I've told you 4 key features of the transcriptional circuitry that we think is critical for programming embryonic cells and iPS cells. This autoregulatory loop formed by the master regulators, the poising of lineage, the genes encoding lineage-specific factors that must be off, that need to be turned on rapidly as cells differentiate. MicroRNAs, we think, tune the messages for ES cell-specific transcripts. Signaling that is actually brought directly to the promoters that are occupied by these master transcription factors via signaling transcription factors. And this thing that I'm becoming excited about, the concept that there are small secondaries, maybe large secondary structure DNA loops that are critical for active gene control in these cells. And finally, that reprogramming in our minds is really, in effect, reestablishing this core autoregulatory circuitry. Perhaps that is conserved in, that type of circuitry exists in most cells of import, and that we may use that information to reprogram cells directly into those cell types once we understand what those master regulators are. The folks who have played key roles in the work that I described are Lori Boyer, who's now an assistant professor at MIT, a group of talented grad students and postdocs who I put on these slides as I showed them, and the work I just described to you was done by, on DNA looping, was done by Steve Bilodeau, Mike Kagi, and Jamie Newman. And much of this work has been done in collaboration with David Bartel, Rudolf Janisch, and Phil Sharp. Thank you very much.